to pull it up on my laptop. Yeah, we are in a new classroom for the first time, and there are a number of students in the room here ready to listen, but unfortunately we can't get it up on the projector, so they all have to pull out their laptops to line up on their own on the Zoom itself. So hopefully we are going to have someone here to help coordinate the technology on this end. But welcome everyone. This is our first seminar of the fall semester here at Vanderbilt University for the Quantitative Systems Biology Center seminar series. And we are very fortunate that our first speaker is an esteemed fellow. He is the James S. McDonald Foundation Fellow at the University of Pennsylvania. And he's also a college lecturer in computer science at the Oriel College and the University of Oxford. He will be presenting today on his work that is using theoretical computational science and applying that to evolutionary biology. Very looking forward to your talk. Thank you.
Yeah, so I'll just share my screen. And I think I've already tested, but hopefully everyone can see. And if I draw, hopefully everybody can see. I drew a smiley face top left corner. Okay, great. And so,
thank you.
As far as I understand, this is supposed to be mildly interactive, but it's hard with Zoom. And so I'll pause throughout to take a couple of shorter questions and then save the sort of bigger discussion questions for the end. I don't think I can monitor chat easily myself, but I'll just ask for questions once in a while, and then if anybody wants to jump in. Yeah, feels a little weird speaking in a systems biology seminar because I'm really an evolutionary biologist, and I feel like a lot of times system biology is more molecular focused, but I think evolutionary systems biology is very important, and it's very important to look at how evolution shapes our organism, and also it's very important to apply systems thinking when we're thinking about evolution. We're trying to understand sort of the power of evolution to, especially in settings where evolution is sort of an adversary against us, such as in cancer. And so kind of throughout the talk, I'm going to focus on that. I'm going to spend probably the first half or so mostly talking about somatic evolution of cancer. So that's how cancer cells are evolving within your body, within a single patient's body. And then at the end, I'll kind of wander off to something that's vaguely related to that, but more sort of long-term evolution. It's only vaguely related because, current research haven't found the clearest ways that it relates to cancer and specifically, but I think it's, all comes from a unified kind of setting. And as was briefly mentioned, my background is actually as a theoretical computer scientist, and I come at these questions from a theoretical computer science perspective, and I try to work from as much as I can from an interdisciplinary perspective and have input from lots of other folks that bring their own different perspectives on evolution, on cancer, on systems biology, etc. And so I want to start with something that Ryan McGee has taught me. Ryan is interested in evolution as a learning process and takes a perspective on, looks at evolution from the perspective of computational learning theory. And one of the ways we can think of evolution is as a game between the evolving population and the environment, where the strategy of the population is the distribution of types in that population. And And the strategy of the environment is the kind of particular challenges that organisms will face in the population. The strategy dynamics for the population are the evolutionary dynamics, and the strategy dynamics for the environment, that's the part we'll be varying to think about various kinds of evolution. And so we can write all of this down as a big matrix, as I did here in front of you. We'll call this matrix, this purple matrix will be called G, and the elements of it will be, Gij. And we can think of the population as selecting a row of the matrix, and the environment as selecting columns, and then the payoff entry is the resulting fitness effect on the agents of that type in the population, right? So suppose we look at type 1 agents, that means they'll be getting a pay, and the environment's in state 1, then a type 1 agent will be getting a payoff of G11, right? If the environment is in state M, then they'd get a payoff of G1M. And this is a nice sort of general model. It doesn't commit us to anything. It lets us think about most things that we'd be interested in thinking about in an evolution setting. For example, by thinking of these things by figuring out how the environment updates its strategy over time, right? So if the environment kind of keeps the strategy the same, it always has the same distribution over potential challenges that the evolving population faces, then that's just a static fitness landscape, right? And we can summarize that as the state of the environment at time t is the same as the state of the environment at time t-1. That's one extreme that you could take. Not the most exciting case when we're thinking about cancer, because in cancer, the microenvironment matters, and that microenvironment is determined by other cells in the population, right? And in that case, we can look at the other extreme and we can say the environmental strategy at time t is actually determined by, is identical to the population strategy. And the way to think of that is basically these individual payoffs can be thought of as what happens if an agent of type 1 interacts with another agent of type 1, for example. Or for this payoff entry that I'm circling right now, what happens if an agent of type 1 interacts with an agent of type M, right? And so that gets into this sort of standard evolutionary game and evolutionary game theory setting. And then, of course, you could think about more arbitrary changes to the environment. And that, for example, is if the environment is controlled partially by, let's say, a doctor. So there's an evolving population and the doctor is trying to change the environmental challenges that the population faces in order to steer the population towards a desired end state to, let's say, control a tumor or something. And so I'm going to kind of talk about all three of these, and I'm going to start with this evolutionary game view of, in particular in cancer, right? So let's focus on the case of where the probability of encountering various environmental states is actually determined by the probability, the proportion of other agents of given types in the population. And this is the standard sort of evolutionary game theory and oncology view. And sort of one of the pioneers in this field is David Basanta over at the Moffitt Cancer Center. And I'm not going to go through the countless work that's been done on EGT and oncology, but I want to pick on one particular paper, one classic paper as an example. This is the go versus grow game, and hopefully it explains better the sort of abstract model that I specified. In this go versus grow game, we can think that there's two types in the population. There are go or invasive cells, so go, or I also call them inv. And then there are grow or autonomous growth cells, AG. Those are the two types of cells in the population. If an invasive cell encounters another invasive cell, so in this picture, let's say, in a given resource spot, then one of them will move over because it's able to move around. It'll move over to find resources elsewhere, and the other cell will stay put and eat the resources that's in that space. So one cell, let's say this cell stays where it is, so it'll end up getting the resource benefit B. The other cell will move somewhere else, paying a cost C to move over, but then receive benefit B of that new resource spot it's found, right? And since the two cells are symmetric, you have a 50-50 chance of being the cell that stays or the cell that moves. And so your overall payoff is down here, 1/2 of B plus 1/2 of B minus C, and which way the dice rolls. And similarly, we could see, okay, well, what if an invasive cell encounters an autonomous growth cell? Well, the growing cell isn't going to move, right? So it's for sure going to stay where it is and get the benefit B, right? So it gets a benefit B when it encounters an invasive cell. Well, the invasive cell will for sure have to be the one that moves. And so it will pay a cost C to move and then get some benefit B at the new spot that it moved to. And then finally, if you think about two autonomous growth cells encountering each other, well, since neither one of them can move, they'll end up having to just share the resource at that spot. So they'll each get a benefit of B over 2. So this is kind of a standard way of formulating evolutionary game in the oncology setting, right? And then, in particular paper, you then go on to link this to particular observations that you or clinicians have been making, and maybe get some insights about what kind of treatments you want to do, or how dynamics are going to develop, etc. What the chance of invasiveness is, things like this. But right off the bat, if you're seeing this game, you might say, okay, but you said that the probability of this happening and the probability of this happening is proportional to the proportion of invasive cells and proportion of autonomously growing cells. But in a real population, cells inhabit space, and so there's like a higher chance of self-interaction, right? If you're a grow cell, you've probably split and you're near other grow cells, and so you're more likely to interact with them than with a go cell. Similarly, if you're a go cell, you might have recently divided, and so there's probably one of your daughter cells around, so you're more likely to interact with another go cell. And you might want to capture this sort of spatial structure, which you can do very abstractly by saying whenever a cell dies, the cells that will replace that cell compete from nearby ones in the space. And those nearby ones get their fitness from interactions with their own neighbors. So let's say the fitness of this cell depends on these pink cells that interacted with, while the fitness of this cell depends on these brown cells that interacted with. And proportional to that fitness, one of these cells will repopulate the spot that perished. And in that case, your proportions aren't, your chance of having various kinds of payoffs aren't directly equal to the proportion of the cell types, because it's not a well-mixed population anymore. There's some structure to it. But if you do your math carefully, and again, you can read the paper for an example of how to do the math carefully, you'll see that this rule, which describes how two cells interact, can be transformed into a game describing how two types of cell interact in this well-mixed setting, right? And this transformation from the reductive game to the effective game absorbs the spatial structure into it. And one of the ways to think about that is to say, in our original version of where we had the game matrix G, which in this case I called it A in this case, game matrix A in this particular case, we had the population versus the environment, but the environment was some complicated function of the population due to spatial structure, right? So that's one way to view it. Another way to view it is to say, actually, there's some other game A prime such that X can be just set equal to P. So it's self-interaction, right? This A prime will be the effective game, this A will be the reductive game, and the A prime absorbs this complicated function. And in cases where you only care about first order effects, or you only care about certain broad features like the difference in the size of neighborhoods of cells, you can make this transformation pretty reliably. And instead of analyzing this reductive game, you just have to analyze a slightly more complicated effective game that's included some correction terms to account for the spatial structure. And so this is kind of a typical use of evolutionary game theory. We start with some intuition about how two cells interact. We combine it with some sort of ideas from, let's say, spatial structure. And from that, we get how the types in the population at the population level are interacting. And then, you might link this to some particular clinical observations, right? You might notice that in a typical tumor, in different parts of the tumor, there's a heterogeneity in the number of neighbors that cells have, number of other cancerous cells that they have as neighbors. So somewhere, let's say, deep in the bulk of the tumor, you have lots of neighbors because all around you there's other cancer cell or standard tissue cells. On the other hand, if you're right up against a membrane boundary, let's say over here, then you have fewer neighbors because some of your neighbors are not really of the same kind as you, right? They're a membrane that you're trying to break through or something. And then even if the cell-to-cell interaction here and here is the same, the fact that they have a different number of neighbors can change the effective outcomes. And so in particular, as you increase the viscosity of the population, or as you, so starting from here, you have many neighbors. This is down here towards the bulk. Up here, you have very few neighbors. That's like a boundary, right? That can transform the type of dynamics that happen, even for the same benefits of resources and costs of moving, right? So for an example, let's say that the cost to benefit in our reductive interaction is 0.53, so this particular line. If you're in the tumor bulk and you have 8 neighbors, then the game will actually select for an all-growing population, an autonomous growth population. On the other hand, that same exact reductive interaction, but happening near a tumor boundary where there's fewer neighbors, let's say only 5 neighbors, you're up against a wall, will now instead select for coexistence of GO cells and GROW cells, of invasive cells and of autonomous growth cells. And that can be very dangerous from the perspective of a clinician, right? Obviously, if you are worried about the cancer metastasizing, you want there to not be invasive cells around. And you might spot some tumor locally, and you might do a biopsy to sample that tumor. Now, as you do the biopsy, you're inserting your needle into the tumor. What's that needle going to get? It's going to get a little bit of boundary, but it's mostly going to get bulk when you suck in some cells, right? And so you'll get that out and you'll put it in your invasiveness detector if you have one. And it'll tell you, oh no, all of these cells are benign, right? Because most of your sample came from here. And so you didn't detect the few invasive cells. But right at the boundary, you could have much more invasive celes because the selection is actually different at the boundary and you're sampling less from the boundary, right? And you can see this clinically sometimes, right? Maybe I have it on a different slide here. If you look at the boundary here, the cells are dyed for invasiveness, and these sort of darker regions are more invasive, and they're all right near the boundaries and stuff. So that's kind of what a typical study in evolutionary game theory and oncology would look like, right? You have this thoughts about the reductive structure of the game. You maybe combine it with some other ideas about the spatial structure or something, and you get some broad conclusions about the effective structure. But the question becomes, how do we know the game? And here, we kind of have to move from modeling to measuring. And here, Jiahua has helped me think about this a lot. She's a philosopher, recently moved from the University of Virginia to Taiwan. And that's, as we move tools, this evolutionary game theory standard story I told you, that's the same as you do with like analyzing gazelles or the sex ratio or something for macroscopic organisms. But in a cancer setting, you get this pressure towards becoming more and more specific and more accurate with your game, at maybe at the expense of generality, because you'll have a different story for each different tumor. And so you want to move from this perspective of modeling, of where you first construct the model, second, analyze, refine, and further articulate properties of that model, and finally assess the fit between that model and the thing and the observed world, to one that's more aimed at direct measurement. So if we look at these three stages of typical modeling, as spelled out by Weisberg, we can see them in our map from the reductive to the effective game, right? We constructed a model about this, about the reductive interaction. We further articulated features of that model by saying, oh, there's some spatial structure about it, and that changes the quality dynamics that happen. We got the dynamic regimes and things like that. And then we vaguely compared it to some images that we've got from, let's say, biopsies or something. But alternatively, we can look at our evolutionary cycle and we can say, well, we know the dynamic of how the population updates its strategy, right? You know, we know that it's following natural selection, that it's following replicator dynamics. And from this, we can actually figure out what problem it's solving, i.e., what is the exact game. And so instead of viewing this as a hypothesis, we can instead do a direct measurement. But maybe, since I was supposed to pause once in a while, maybe I'll pause before I go on to direct measurement. Is there any questions about this sort of typical approach?
I do have one question from Heather Hartman. How do oncologists then use game theory in the clinic to help with analyzing cells or biopsies?
They tend to not use it in the clinic because they don't know exactly what the game is. It tends to be theoretical work that sort of suggests new things that clinicians should think about. But then for the direct applications so far, it's just clinicians now think about that. They might then do a new clinical study, and then that work proceeds as it normally would in a clinical setting. So it's typically more of a hypothesis generating mechanism as a theoretical grounding for some further work. But we're trying to change that. We're trying to change that by giving specifically how can we measure these games in a particular patient and then use that to inform treatment. So that'll actually read back to this section of measurement. So let's move to this measurement. So how can we directly measure this game? How can we make a game assay? If we're doing it in the simplest place where we can start, which is in vitro, we can imagine having our 5 beakers and 2 cell types, and we'll fill one beaker all of the red cell type, and 1 beaker all of the blue cell type. 1 beaker will be, let's say, 50-50 of the 2 cell types, and this beaker will have mostly red, a little bit of blue, and this beaker will have mostly blue and a little bit of red, right? We can set up an experiment like this. And then in this experiment, we can then use that as our x-axis, that's our control, right? That's the proportions that we seeded of blue from 0 here to all here. And for each one, we can measure the fitness. We can measure the fitness of the blue cells, They're expressed as blue dots here, and we can measure the fitness of the red cells. Again, in this cartoon, they're expressed as red dots. Then from this, these are of course discrete measurements because we can only do so many different combinations of proportions as experimentalists. And of course, they also have error bars, etc. Just skipping those out for the cartoon. Based on those, we can now fit a fitness function, right? And we can say, based on how these dots are lining up, it seems that the fitness of the blue cells, which I'm going to call the C cells, is something like this, while the fitness of the red cells, which I'm going to call the D cells, is something like this, right? Now I have those two fitness functions. Based on where those fitness functions intersect P equals 0 and P equals 1, I can recover the corresponding evolutionary gain. And so this experimental procedure of seeding certain proportions initially, getting their corresponding fitnesses, and then figuring out what kind of function describes the relationship between proportion and fitness gets us, as a result, this thing called the effective evolutionary gain. And so from that, we can figure out things like the gain function, what's the chance, what's the advantage of the C cells versus the D cells. From that gain function, we can look at the where the gain function intersects the P equals 0 and P equals 1, and that gets us coordinates for a game space. And we can say, this game is right here in game space. And so we can change this big array of measurements into a point in game space. And so this is the kind of things that we've done now experimentally with non-small cell lung cancer. So we consider 4 different conditions, right? Two different cell types, the drug-sensitive cell types, which I'll call parental here, and the drug-resistant cell types, which I'll call resistant here. We might look at the fitness functions for them in, let's say, drug, or we can look at them in fitness functions for them in the condition where there's drug and also some cancer-associated fibroblasts around, right? Or we can look at the fitness functions in, let's say, no drug setting or no drug, but the cancer-associated fibroblasts are still around. And from this, we can get the 4 gain points corresponding to those 4 different conditions. And in this particular case, these 4 gain points end up being in two qualitatively different regimes. So they all differ from each other quantitatively, right? You can see none of the error bars of them overlap. So they're all quantitatively different from each other. But most importantly, they lay in different quadrants of this game space. So the DMSO plus CAF condition, which is kind of the condition corresponding to a typical pre-treatment patient, lays in this upper right quadrant, while all the other conditions, which is setting where the supportive tissue was eliminated or where we started treating with drug, are kind of in this bottom right quadrant. Those have relatively different dynamics. In particular, in the top right quadrant, your dynamics push you towards coexistence of parental and resistant cells, or of resistant sensitive cells, while in this lower quadrant, they push you towards all resistant cells. And of course, you can this is a particular case that we did in 2019 or something. I think this was the first time such gains were measured directly. But you can always move this on to you can move from 2 to 3 strategies. So for example, Ranjini has, we know that HGF is very important to this interaction. So she's focused on, how does the HGF change the game? And so you can look at the game payoff of sensitive versus resistant cells that we were talking about before and expand the strategy space. Right now, sensitive resistant is a 3rd type of HGF producers. You can measure those pairwise payoff functions as you did before, and you can recombine those into sort of a 3 strategy, more complicated setting. When you move from 2 to 3 strategies, you get much more a richer possibility of dynamic regimes. I won't go into it because I kind of want to move on to some other things. Similarly, you might say, okay, well, that was one drug that we looked at. We looked at how alectinib changed the game that was happening in the tumor. We can look at, a richer set of drugs. So Nathan has done this to get an idea of how much competitive release there are to different drugs. And again, I won't go into the details of the difference between sort of absolute fitness competitive release versus relative fitness competitive release, and just get to the idea of, when you get to a game space, you can sort of imagine this big game space of possible games. And for each one of your drugs, you can imagine them as a point in this game space, right? And then depending on what part of the game space they're in, they have different features. So in this particular case, these drugs down here have the possibility of competitive release. And so that with these drugs, things like adaptive therapy might work, while with drugs sort of above this line, there is not competitive release. So trying to do some sort of adaptive therapy with these drugs is probably not going to be successful. It's probably better to do sort of maximum tolerable dose type treatments with those drugs. And so that's how we can imagine this in the future informing based on some assay out of the game that's describing what's happening in a patient's tumor, adjusting what kind of strategies the doctor might try with these drugs. Obviously, at this point, it's far too very far away from the clinical setting. I would never move this to the clinic, but we're kind of getting started at this research direction. So is there any questions about this general idea of assaying the game that describes the interaction within a tumor?
We have one question from Jason Hughes. For getting direct measurement of evolutionary gain by varying the seeding, does how mixed the population is in the local structure or their spatial heterogeneity or degree of mixing strongly affect the fitness for each population?
Yeah, it does. And that's why this measurement is a measurement of the effective rather than reductive gain. So in that story we had before, where in the go versus grow story, where we had a description of what happens with two cells interacting, and then spatial structure changed that into a population level description. So this assay is measuring just the population level description. And that measurement thus combines both the cell-to-cell interaction that happens plus the effects of population structure, like spatial structure, into a single measurement. And in the case of worrying about what's going to happen to the patient, that's really all we care about, right? We don't really care about exactly what's happening as one cell talks to the other cell. We really care about, is the tumor going to grow? Is the tumor going to shrink? We care about these population level properties. And so in that case, we don't necessarily need to push down lower, but future work, some of the future work that I'm trying to do with this is also figuring out, once we have this population level measurement, and let's say we have some information about the spatial structure, can we push down through the spatial structure and figure out what the local interaction is like? That hasn't been done directly yet, but we're kind of working on that with some collaborators.
Awesome.
Is there anything else on the game assay?
That's all we have for now.
Okay, great. And so the reason you care about knowing these games, and in general why you care about knowing sort of how drugs affect the fitness of cancer cells, is because we imagine that as clinicians, we'll make decisions based on that, right? And this moves us to this view of treatment that Katerina Stankova has been sort of pioneering, of viewing the challenge faced by it depends whose side you're on, the tumor or the doctor, as this game between the tumor and the doctor, right? So the state of the environment is not controlled just by the tumor and the distribution of strategies, but also by the doctor choosing which drug to administer. So an example with the 4 games that we measured in non-small cell lung cancer, then the big environmental state matrix. And you can imagine, my internet is unstable. Can everyone still hear me?
We lost you for a second, but I think you're good now.
Okay. We can imagine them all as part of this big purple game matrix, and the clinician can select which of 4 different strategies to employ. But they only have partial control. They can't pick a single row, a single column to play. Instead, they can pick a sub-matrix to play. So there is some constraints on what the doctor can do. And in the particular case of this game, of this environment, no matter what the doctor does in our system, it's not going to be good for the patient, unfortunately. But you can imagine with better drugs, you might have a combination of strategies the doctor can do that will help the patient. And this is one of the things that we have to worry about. From the perspective of computational learning theory, replicator dynamics as a way of updating the strategy of the population is actually an optimal dynamic for playing against sort of anything the doctor can do, all right, in some very precise sense, which I won't go into the details of. So that means we shouldn't underestimate the tumor as sort of not good at what it does. The tumor is actually very, very good at trying to maximize something that's roughly equivalent to tumor size, not exactly, but roughly equivalent to tumor size, relative lineage fitness in particular. On the other hand, the doctor does have various handicaps in that they can't do certain kinds of combinations because certain drugs aren't available. Or when you give a drug, there's like subtypes that resist that drug, for example. And so how should we as clinicians respond to this based on this knowledge? The standard approach has been, let's make new drugs that change the game matrix such that even though the tumor is playing optimally, even the best it can do is bad for the tumor and thus good for the patient, right? That's one way we can do it. Another way we can do it is this sort of thing that's been popularized a lot with adaptive therapy, which is, okay, well, we have the drugs we already have now. Can we do anything given this? Well, we can pick which drugs to give in which doses at which time. So from the perspective of this framework, we can try to mess with how the X vector depends on the P vector in hopes that we can kind of steer the tumor where we want to go. But again, we're trying to still play against the tumor at the game that it's good at, which is at sort of reducing, the tumor is good at maximizing its size, and we're trying to reduce size. We're trying to beat it at its own game. And when you are in the setting of where you're not particularly good at a game, let's say when I was a kid, I wasn't very good at soccer. And so what would happen, I'd lose games, but then my parents would say, oh, don't worry, it's not about winning the game, it's about having fun, right? So what's the parent doing there? They're changing what the goal of the player is. They're making the goals different for the two players, right? Don't make it about winning the game, make it about having fun. And we can do something similar in the case of treatment of patients, right? We can change our goal in the game. Instead of trying to focus on minimizing or controlling tumor size, which is something that the tumor is really good at as an adversary against us at, we can instead focus on controlling the negative effects of the tumor on health, right? So a priori, a large tumor doesn't mean bad for patient, right? If there's ways to drive the tumor, for example, to make tumors more benign, right? So you treat with drug, not with the hope of reducing the tumor size, but with the hope of making sure the tumor doesn't metastasize. Then you might still have a relatively large tumor, but one that doesn't ever spread and doesn't threaten the patient's life. And so, this is the kind of perspective that thinking about the relative power of the evolutionary player and how they're pretty good at the game can change our perspective on what we should do as clinicians. Does anyone have any questions about this tumor doctor game?
Well, we have one question in the chat from Chen Wang. For the evolutionary game overall, are there any notable differences between various ethnicities?
So in our experiments, this was done in vitro from one specific patient's derived tumor. So we didn't test, not only did we not test across like different ethnicities, we didn't even test across different tumors in different patients. This was sort of the first time the games have been measured, and they were measuring a particular patient to be this particular thing. But yeah, the hope is that once we can do these things kind of effectively, and once we have some reason to believe that the kind of measures we do are clinically relevant, then you might want to move on to, looking across different tumor sites in the patient, looking across different patients with similar tumors and how much translation there is there and how much personalization there is there. I wouldn't know. Based on what we've done so far, we can't say anything, but I would expect it to be different.
Awesome, thank you.
Great. So far, we've been talking about this game between the population and the environment, but the number of types in the population was sort of low, right? I was showing you 2 types, and I talked briefly about Rangini's work, which had 3 types. I didn't really go into detail of it, but that's not what happens in the real world, right? That's what happens in a well-controlled laboratory where we can get just 2 types of cells and we can color code them with, green and red fluorescence. But in a real patient, or if we're looking at evolution, not even cancer evolution, but evolution more generally, one of the things that starts to matter a lot, one of the things that as systems biologists we should care a lot about is how are new types generated, right? So we know that the evolving population is very good at, once there are certain types in the population, it's very good at choosing the best types among those, right? So natural selection works really well, but that's not what matters in the very, very long term. In the very, very long term, what matters is how do new types enter the population? And so now we're imagining that this matrix has very, 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 very many rows. Well, in that case, let's just focus on this mutation generating new genotypes. So how are new types introduced into the population? And let's not worry too much about the ecology of the interactions, right? Now let's divide our problem as, there's some algorithm of how are new types generated in the population, which we might not know the details of because we don't have great knowledge about the sort of on how even at the genotypic level mutations look like. But more importantly, on the phenotypic level, we don't have a great idea of the genotype phenotype map. And so we really don't have a great idea of the algorithm of how new variants are generated in the population. But maybe we can have a good idea of the fitness of the various variants in the population. So if we know this, can we abstract over what we don't know here? And so in this case, let's move to the simpler setting to start, which is a static fitness landscape. So moving away from the evolutionary game towards a setting where the environment is going to be the same always. right? So think of something like Lenski's long-term evolution experiment. We're trying to get the same environment for the population, for the evolved population over time. And in this case, people imagine landscapes as this left one often, right? They imagine a landscape where there is some peak and the population quickly moves towards that that local peak. And one of the things that I've shown recently is that that's not really all that's possible. There's this idea of hard fitness landscapes, which, even though the population is always moving uphill, it takes exponentially long for it to find any local peak. It just keeps winding around, right? It's not that some peak is blocking it from going to a higher peak. It's not even finding a single peak is not possible for it in any reasonable amount of time. I basically have to like try all the types before it finds even any local peak. And you can imagine this as a cartoon, as a sort of winding spiral that I drew here. But obviously that's the whole point here is that cartoons in 2 dimensions mislead us about high dimensional genotype spaces, and we shouldn't be using cartoons. Instead, we should be looking at sort of more abstract fitness landscapes. And what makes fitness landscapes hard in the general case of high dimensions is epistasis, interactions between genes at different loci. In particular, we can think of, a little bit off here, but we can think of 3 different kinds of epistasis, right? So if, regardless of the background, the direction of higher fitness is the same. So in here, if the second loci is, the second gene is little b or the second gene is big B, it doesn't matter for the first locus, it still prefers big A over little a. So fitness goes this way, and same here, big A is preferred over little a, right? And vice versa for B, regardless of what the first locus is, little a or little or big A, the second locus prefers big B over little b, right? So this would be an example of no epistasis or just magnitude epistasis. On the other hand, we can think about more complicated things like sign epistasis. So for sign epistasis, the sign of the fitness effect changes based on the background. So here we can see that in the background of the second locus being set to little b, we prefer big A over little a. On the other hand, if the second locus is at big B, then we actually prefer little a over big A. So we have a sign change in the fitness function. That's called sign epistasis. And then finally, there's this idea of reciprocal sign epistasis, which is sign epistasis on each of the 2 loci. And this roughly translates to different types of fitness landscapes, right? So reciprocal sign epistasis is required to have multiple peaks. So usually, if you have a rugged fitness landscape, you have to have reciprocal sign epistasis somewhere in the landscape. If you have a smooth fitness landscape, then it can only have magnitude epistasis, it can't have sign epistasis. And then I introduced this idea of semi-smooth landscapes, which have a single peak, always have short paths to the peak, but they have sign epistasis that blocks some paths. And based on this, you can get different results for the hardness of reaching local optima in these landscapes. So on a smooth landscape, it's always easy for sort of any uphill dynamic to get to the local peak. For semi-smooth landscapes, it's hard. It can take exponentially long, even if you're going up the steepest incline, or even if you're picking a random uphill step. There's landscapes that have a single peak, and yet it takes exponentially long to find that peak going uphill. And then these landscapes kind of look like this, where you kind of end up, you're trying to get from here to here, but instead of finding this quick path, if you follow the steepest gradient, you end up kind of going all the way around and looping around forever for a really long time before getting to the peak, right? You get these kind of winding landscapes. And it can get hard for, if you're in a rugged setting where you have reciprocal epistasis, this can become hard for sort of any evolutionary dynamic, even ones that don't follow up hills, even ones that go downhill and jump around on the fitness landscape. It can still be hard, assuming some results in computational complexity hold. And this can be translated into results about the fitness trace. It can mean that if we don't care about the exact peak, but we only care about getting close to it, then we can always achieve sort of power law drop-offs in the selection coefficient. But on hard fitness landscapes, we cannot achieve an exponential drop-off, an exponentially quick equilibration, right? So if you're familiar with sort of typical, physical systems, and in typical physical systems, you approach equilibrium exponentially quickly, right? That would be an easy landscape. But on hard landscapes, that can't happen. You can only approach the local peak with sort of a power law. And you get this kind of thing happening experimentally in things like Lenski's long-term evolution experiment, where the drop-off in the selection coefficient looks like a power law instead of an exponential equilibration towards something. And if you want to look at the work, you can, from the math perspective, you can look at the constructions for this. Is there any quick questions about the idea of a hard fitness landscape?
No questions for now.
Okay, great. And so in that, we were looking as, is there any occurrence of reciprocal synapses? Obviously, a single occurrence of reciprocal synapses somewhere in the landscape is going to be a big deal, right? Reciprocal synapses being at every single locus everywhere is also not going to be a problem, because then there's going to be so many local peaks that it's always easy to find one. There's some structure to the epistatic interactions that make these landscapes difficult or hard. And we can represent that structure of epistasis as a gene interaction network. We can look at our whole fitness landscape, and if we see, this type of, let's say, fitness, so in green here we have the fitness payoffs for these 4 genotypes. We could write that down as a gene interaction network between the X1 locus and the X2 locus. And so where X1 picks the row and X2 picks the column. And so when 0, 0, they pick 1, 1. When it's 1, 1, they pick 3, right? And similarly for the other ones, right? And this case, the interaction is kind of pointless to write. You can actually eliminate it. It's the same thing as writing down this gene interaction network, which is uncoupled, and that's because it's the smooth landscape. But you can also make the same things between various gene interaction networks that have sort of superfluous magnitude only interactions, and you can always reduce that to sort of sign equivalent networks that have the same direction of arrows, but maybe not the same exact fitness functions. And based on that, we can ask, what do these gene interactions look like for easy versus hard fitness landscapes? And here you can sit down and prove that if your gene interaction network is tree structured and biallelic, then it's always easy. You can always find a local peak within about n squared number of steps at most. On the other hand, once you move to sort of from biallelic to triallelic systems, or when you move from gene interaction networks that are trees to gene interaction networks that are, have tree width 2, then you can no longer make these guarantees, and you can start to get hard landscapes. And from this, you can kind of get an idea of what the next steps are in terms of understanding the boundary between easy and hard landscapes. There's a lot of theory to be done, which is sort of proving exactly what features of gene interaction networks make a landscape easy versus hard. And then you can combine that with experiments where you take real experimental systems, do a local measurement of the fitness landscape, because you can't measure the whole landscape, right? It's exponentially large. You can't do, 2 to the 60 measurements. But you can do a local measurement, and from that infer a gene interaction network, and then figure out where does that gene interaction network fit into your hierarchy of hard versus easy landscapes. And if it fits towards the bottom of the hierarchy and it's a very relatively easy landscape, then you can say, oh, in this case, we can make these assumptions about systems equilibrating quickly we don't have a moving target, we're going to quickly find local peaks and have more or less stable type composition in the population. So, from the perspective of something like cancer treatment, that would be a simpler system to potentially treat. On the other hand, if we measure these and it ends up being very hard somewhere up here, we can say, oh, in this system, we're probably not going to get to local peaks, which means the population will always be evolving. And that might change our ideas about how we approach treatment or how we approach this particular evolutionary system. But these are all next steps in terms of how to link some of this theory to the sort of experiments we might be able to do in either cancer settings or in other natural systems. Anyways, thank you all for listening, and I'm happy to answer any questions either in the 3 minutes we have now or in the hour afterwards with students.
We don't have any questions right now, but if anybody has any, feel free to drop them in the chat box or the Q&A box at the bottom of your screen. I see Darren got the projector working.
Well, we've got us from the crowd in here. Everyone's thanking you for the presentation. I actually had a whole bunch of questions. I'm going to ask one before the hour is up. I'm curious that your whole theoretical framework for the different landscapes, fitness landscapes, and how you put in the perspective cancer. Obviously, there's a lot of genetic heterogeneity. So I'm wondering how you view the genetic effects on the landscape and how it would differ from, say, one cell to the next if one has a genetic alterations as another. Does that make the whole thing change, or are there subtle differences, and you compare them with any of the tools that you've been looking at?
Right. So for in the particular case of cancer, I probably wouldn't approach it as sort of static fitness landscapes to begin with, because we just know that the microenvironmental interactions matter so much. And so you'd need to move more towards sort of a game landscape perspective where you have sort of exponentially many possible strategies. Only some polynomial fraction of them is present in the population, but they also have some interactions coupling between them. And there isn't much work on that kind of, like not much rigorous work on that kind of setting. Obviously, those can express the static fitness landscapes as a special case, and so the hardness results still carry over. There are these weird landscapes that will have sort of optimization happening forever. But in the non-static case, it's actually easier to get these results, right? If you take like a rock, paper, scissors game, the population will always be moving away from you as rock replaces scissors, and as paper replaces rock, and as scissors replaces paper. So I think this open-endedness and endless evolution is actually sometimes even easier to achieve in a cancer setting with even few types.
Thanks, that's fantastic. We are right at the hour and haven't seen any other questions.
I think Jason has a question, but is Jason able to attend this discussion in a moment?
Yes, we can follow up.
Okay, that'd be great.
So let me, I have to hop on my other computer. Everyone is, all the students and postdocs are welcome to join for the ongoing discussion, but we'll have to hop off this link because the webinar format makes it much harder to have this interactive session. So we're going to jump onto the other link that I placed in the chat, and we'll give the speaker just a couple minutes to take a break and meet him over in the discussion afterwards. Thank you very much. Appreciate it.
Okay,